Hey Team Bio, welcome to your screencast on cell size. Today we're going to be answering the question, why are cells so freaking tiny? Okay, so cells. On average, we see that prokaryotic cells are between 1 micron, that's a millionth of a meter, and 10 microns. And eukaryotic cells are between 10 microns and 100 microns. So even organisms, like if you imagine an elephant, elephants are enormous, but if you look at their cells, they're no bigger on average than the cells in your body or the cells in your dog's body or cells in a fruit fly's body. Prokaryotic cells are a little bit smaller. They don't have a nucleus and the same number of organelles inside of them. And all of these organisms, even though they can be quite large, their cells are around the same size. So we're answering the question today, why are cells so tiny? Even a giant amoeba requires a microscope to see. So what controls the upper limits of cell size? And the answer is the laws of thermodynamics, physics, and a little bit of math. Okay, so you eat food. That food is ground up in your mouth and you begin chemical digestion in your mouth. It proceeds through your digestive tract and then it has to be absorbed across the intestinal wall. And every single cell in your body is getting the nutrition it needs through its own membrane. And you get rid of waste. You exhale some waste in the form of CO2. You poop out some waste in the form of undigested material, and you pee out some waste that your kidneys have filtered through your blood. But all of that waste came from cells that needed to get it out through their membranes. So I guess the summary of that is cells get all they need, get the materials they need to survive by moving stuff through their membrane. Okay, oops. Okay, so we know the different ways that things move in and out of the membrane. And the simplest way that material moves in and out of the membrane is through simple diffusion. This is when small, nonpolar or uncharged, and it has to be both, things move directly through the membrane. So an example of this was we had oxygen and CO2. Now oxygen is constantly being produced. No, sorry, that's wrong. Don't write that down. Oxygen is constantly being consumed in a reaction that occurs in the mitochondria. So if you can imagine, if you looked at a cell, you would see a greater concentration of oxygen outside of the cell than you would see inside of the cell, because it's always being consumed inside of the cell. So oxygen diffuses down its concentration gradient, and when it reaches the mitochondria, it is consumed in this reaction cellular respiration that we're going to talk about later. Now, at the same time, glucose is constantly being cut up into small, small chunks of CO2. And that process is powering all the reactions that happen in your body, really. And so you have a really high concentration of CO2 inside the cell right next to the mitochondria because it also diffuses down its concentration gradient. So remember, these small nonpolar molecules can move directly through cell membranes. So they move from an area of high concentration to an area of low concentration because that's what the laws of thermodynamics say they must do. They are trending towards thermodynamic equilibrium. Okay, so imagine we have a small cell. Things need to get in and things need to get out. And we have a much bigger cell, and the same is also true. We have oxygen that's in high concentration outside of the cell and a lower concentration inside of the cell. Very little oxygen in here. And CO2 in high concentration inside of the cell and low concentration outside of this cell. Okay, now you all know that the rate of diffusion, how fast these little molecules are moving, is dependent on the temperature. So let's write some of these things down. Okay, so these are small uncharged molecules, and the rate at which they diffuse is dependent on the temperature. Okay, so that means that if we look at these two cells, it's going to take a lot longer for CO2 to diffuse out of this guy and oxygen to diffuse into this guy than it is for CO2 to diffuse out and oxygen to diffuse in of the small cell, just because this has a lot less distance to travel than this one. The only way we can make this diffusion happen faster, oxygen diffuse in faster and CO2 diffuse out faster, is to increase the temperature. So if we increase temp, then faster diffusion. However, as we keep increasing the temperature and getting faster diffusion, increasing the temperature, faster diffusion, this other thing is going to happen where proteins denature. So here we've hit a little stumbling block. This cell can't get any bigger because diffusion is happening at a fixed rate, but at a rate that it can't get the things that it needs fast enough inside the very middle of its cells and the things that the waste products that it's producing out fast enough because they just have too far to go. And it can't get any warmer because then all its proteins start to denature. They start to unfold and not be able to catalyze their reactions. Okay, so simple diffusion and the fact that it's dependent on the temperature and the temperature can't get too hot inside cells is maintaining cell size to be small. Okay, another reason that cells stay small. Okay, let's call this category membrane transport. And in this category, we have protein-mediated passive and active and bulk, which includes endo and exocytosis. Okay, again, all of this movement of materials happens through the cell membrane, but this type of membrane transport is a little bit more regulated than simple diffusion. Okay, So this is all movement through the membrane that is not simple diffusion. But just like simple diffusion, these things occur through the cell membrane. And now's the time that we're going to do a little math. Okay, we are going to calculate the surface area to volume ratio. So that means we're going to look at the surface area of this cell and look at the surface area of this cell, and we're going to compare the surface area of the cell to the volume of the cell, and the surface area of the cell to the volume of this cell. So first, we have to calculate the formulas for surface area and volume. So the surface area of a sphere is equal to 4 pi r squared. And the volume is equal to 4/3 pi r, I'm going to use the same r, pi r cubed, where r is the radius in both of these cases, the radius of these two cells. Okay, so if we want to calculate the surface area to volume ratio, we put the surface area over the volume. So we can just now say that we have 4 pi r squared divided by 4/3 pi r cubed. Okay, now we do a bunch of fancy math. The pi's are going to cancel out. Let's do that. Pi's gone. The 4 is also gone. 2 of the r's are gone, leaving 1 r below. Oops. So, and the 3 is, we're dividing by this, so now it's going to be on the top of the fraction, the numerator. So we're left with this equation, which is surface area, surface area to volume ratio is equal to 3/r. What does that mean? Let's interpret it and write it maybe in a new way. So, obviously, this cell has a smaller surface area than this cell. So, let's write, as the radius increases, the surface area increases. Obviously, this little cell, has a smaller volume than this cell. So, also, as radius increases, the volume increases. However, as the radius increases, I suppose I could have just said that once. The surface area to volume ratio decreases. So let's look at this equation to find out why. Now, imagine the surface area, or the radius of the cell is getting bigger and bigger and bigger. And here we have our equation for our surface area to volume ratio, 3/r. So imagine the radius is 1. Now, this ratio is just equal to 3. So the surface area to volume ratio is equal to 3. But now let's imagine making the radius bigger. Now the radius is 3. So the surface area to volume ratio is 1. It's gone down. Now the radius is 10. The surface area to volume ratio is going to be 0.3. It continues to decrease as the radius increases. So what does this mean for the cell? So imagine taking a piece of the cell membrane in each cell. So let's take this little green slice here and this little green slice here. And we're gonna tunnel a pyramid from this square that we're taking of the surface area here down into the center of the cell. And kind of like slicing a three-dimensional pie. I guess that pie is three-dimensional. Kind of like slicing a spherical pie, which would be very hard to do. So you slice your spherical pie and you pull out this wedge of surface area with the volume down to the center. And then you do the same thing on this size. So you remove it from the small cell and from the large cell. And the same piece of membrane, so this is the same size membrane here as here. The same piece of membrane has a much larger interior slice of pie in the big cell than it does the small cell. Now, if you're talking about pie, this is a good thing because you have a giant slice of pie that you get to eat. But if you're talking about feeding the cell, it's bad for the cell because this means the big cell needs more stuff. It needs more food. It needs to get rid of more waste. It needs to feed this entire slice of interior volume with this surface area. Whereas the small cell only has to feed this much interior volume for the same slice of surface area. So for the same corresponding piece of the membrane, the larger cell has much more body to feed, and waste to remove than the little one. So that's why it's such a big deal that the surface area to volume ratio decreases as the radius of the cell increases. Cells can't feed themselves. Okay, and that is the upper limit on cell size. So the rate of diffusion and the surface area to volume ratio determines how big cells can get. Okay, this was kind of a long screencast. I apologize.